Good evening, everyone. I would like to tell you about the biological research in space era. So let's start. When every living organisms leave the Earth and go to low Earth orbit, what happens? These organisms face the different conditions, which include microgravity, cosmic radiation, vacuum, extreme temperatures, and closed spaces. I'm sorry. The first space flights with living organisms started between 1940s and 1960s, and the main idea was to understand if a human can survive in these conditions. So the first astronauts were fruit flies, mice, bacteria, dogs, plants, turtles, and even monkeys. So these experiments showed that all these organisms can survive and can be back to Earth safe. Even monkeys can save their cognitive functions during the orbit flight. So the next step was to send Yuri Gagarin to space. As all we know, I'm sorry. From 1961 till now, more than 600 astronauts visited low Earth orbit and even Moon. Then more than 300 experiments were conducted on human health in microgravity conditions. So what we know for now, what happens to human body in the space during the space flight? One of the most severe problems, it's spaceflight osteoporosis. The second one is cardiovascular disease, radiation-induced DNA damage, vestibular disruption, vision impairment, and weakened immunity. In bold, there are the most severe problems, which we will discuss further. First is the bone mass lost. Its rate is 10 times higher than for old people on the Earth. It means that astronauts lose from 1 to 2% their bone mass for months. So if they spend, for example, a year in a space, they can lose about to 20% of their bone mass. And it's a very, very high rate. And the one side, it's a good model to discover new drugs and to test them. But the bad thing is for astronauts, their recovery after the space missions, it's very hard. They can break their legs, and it's very, very bad. So what can we do to prevent this? There is now developing a lot of special drugs to prevent bone mass loss. And the most promising candidate is the laboratory engineered protein based on original protein, which is responsible for bone formation. It was already tested on the orbit, but not on people, on mice. These mice got injection of this protein for every 2 weeks for 9-week space mission. And the result showed that it significantly increased their bone mass. It really helped. So this promising drug called BP-NOPEC, and it will be tested on human then. We are waiting for phase 1 human trials. The second problem is weakened immunity, and we will talk about a little more. What we know about human immunity itself? It consists of 2 main parts. First, it's real human cells, different one, which called neutrophils, macrophages, T and B cells, and natural killers. All of them suffer in spaceflight conditions. So the human body faces the problem with reduced ability to fight infections, more inflammation, impaired response to vaccination, increased risk of chronic disorders and even cancer. And the second part of immunity is human microbiome. In our gut, we have special microbes which protect us So during the space flight, this microbiome also feels stress, and the balance is changed. So we have to do something to save this balance. And there are some ways to do it. First is probiotics, which are used on Earth too and on the space orbit right now. And there are future plans to do something like ideal human laboratory engineered microbiome adapted to spaceflight conditions, but it's still a dream. Second, more simple variant is to frozen preservation of original microbiome of astronauts and regular supplies of original microbiome, but it's like not really good solution. Laboratory engineered microbiome would be a greater, but not now, maybe in 10 or 20 years. And the last outcome of weakened immunity is viral reactivation. What does it mean? There are some viruses that live in our body all the time. Almost all people have, for example, herpes virus, which is sleeping in our nervous cells. But when our immunity is weakened, this virus starts replication, and so we get ill. What does that mean? When astronauts go to orbit, they're pretty healthy, but on the stress conditions, their latent virus can reactivate. So there is no way to eliminate all these viruses from astronauts' body. So we have to find a solution for this process. What can we do? First thing is make regular monitoring. We have to monitor two things. First is blood analysis on board of International Space Stations to check the status of our immune cells. And the second is antiviral system testing. So if there is something viral reactivation, or maybe there are some infections on board of International Space Station or other spaceships which will be created further. And the other countermeasures include physical exercise, which are already used on board, maybe artificial gravity, which will help to make space conditions more looks like on Earth, development of new drugs with anti-inflammatory effects and immune stimulants, and create new radioprotector drugs. We will talk about them a little bit further. The next thing is using probiotics, which are already used, and development of new vaccines, which will work better on the space conditions than now, and boosters for long missions. On these pictures, you can see real tests of a blood analyzer. These tests were performed in 2020-2021. This simple machine works like a real blood analyzer. I will show it in the next slide. It's like this one size box, not a really big box. And this box can, in 3-5 minutes, show the immune status of astronauts. It works this way. We need just a drop of blood of astronauts, put this blood on a disposable cartridge, this one on the left picture, and then in 3-5 minutes we have the result. If on board there are people who understand how to interpret the result, so they already know the result. If not, they send it to Earth and then get the definition what is happening. So really, for now, astronauts already can do regular monitoring of their immune status. The second monitoring system, which was already tested on board of International Space Station, it's a rapid pathogen detection using lamp amplification. I will try to explain what is it. This looks like a test for COVID-19 PCR test. I think every one of you did them. This technology is kind of that. It's based on the bacterial system of DNA replication. When bacterial cell starts to divide, first step is to replicate the DNA, the genetic code. And for replication, bacteria use special enzymes. So people took these enzymes, put in the tube, supplied all necessary salts which these enzymes have to use. And then we put there our sample, purified one, for example, sample of blood or sample of air, water, or something you want. We heat this tube for 65 degrees of Celsius for 30 minutes, and then if color changes, we see the result is positive. It's the simplest for usage, the simplest system which was developed for usage in space conditions. In our laboratory, we have already also PCR machines, but the protocol it's much more complicated. Here you see real pictures from on board of International Space Station. The yellow color in the right side tubes show that this experiment was successful. But the problem is there is no protocol for purifying the sample on board still. So this system was tested by purified samples from the Earth. Because sample purifying needs several steps of liquid mixing. And this is a problem in the conditions of microgravity. So the system is still on development. But it's a big step forward to monitor what is happening on the International Space Station. The next thing, what happening with human, it's DNA damage caused by radiation. And it's also a difficult thing. It causes the increased mutation and cancer risk. So what we can do with that? First steps are technological, like engineering. We need to develop shielding for clear models, more effective one, and monitor solar flares to not to be exposed to higher doses of radiation. And the thing we will discuss, it's research of radioprotective drugs. So how can we research them? Let's start from the beginning. How DNA damage occurs. The DNA, it's a double helix, and each strain consists of 4 types of monomers. They're called nucleotides. There are 4 types: guanine, cytosine, thymine, and adenine. And the sequence of these nucleotides hold all information about our body structure and how our body has to work. And there is one rule that you should remember. If we have on the one chain cytosine, on the same place on the other chain we will have guanine. And if we have adenine, so on the another chain we will have thymine. What does it mean? It means that the information on the one chain is the same information that on the other chain. On this basis is placed all cell dividing. So when a living thing is exposed to high dosage of radiation, this radiation makes the reactive oxygen species from the water inside of the cell, and this reactive oxygen species physically damage the DNA chain. It could be one strand break or double strand break even. Then, because the cell is not stupid, it can protect their cells with special repair enzymes. But if the rate of damage is very high, so the repair enzymes cannot fix all of them. That's why we have mistakes. And if we have mistakes in DNA, so all other cells after dividing of DNA with mistake, all these cells will have the mistake the same. So all daughter cells will be mutated, and if it happens in germ cells, so we could have children with mutation. So it could be dangerous. As illustration, I can tell you about the one research by about astronauts, which had a flight between 1998 and 2001. This flight was just 12 days long, and after these 12 days, scientists took peripheral blood and purified DNA from this blood and sequenced the genome of astronauts. Why they took the blood? Because blood cells, they renew every several weeks. So this blood was just freshly synthesized. And what have they found? They found that for 14 astronauts, there were 34 mutations. The most of them were safe, but some of them were bad one. They are colored. There just were a few of them, but it was just for 12 days. So imagine if we spent on the Earth, on the orbit, for example, not 12 days, but a year. So the rate of mutation could be much more higher. That means that we need really good protection. To protect ourselves from DNA damage, we have to look at organisms that already have this superpower. One of these organisms is tardigrades. Also, they called water bears. Do you know what is tardigrades? Please raise your arms if you know. Oh, not all of them, so it will be a surprise for you. Okay. It's tiny creatures, like less than 1 millimeter, and they live in almost everywhere. And they can survive extreme conditions, like 5 kilograys radiation exposure. It's about 1,000 of human lethal dose. Also, they can replace more than 90% of their water by special sugar and become a state of a tune. It's like a sleeping dormant stage, and they can stand there for years. And in this stage, they can survive even open space condition. They spend there 10 days, and more than half could survive after that. So what is their secret? One of their secrets, one of the ways how do they protect themselves, it's a special radio-resistant protein. It's called damage suppressor or DSAP. It's special protein. It's synthesized when tardigrade face the stress, and this protein goes to its DNA, and this protein just hugs it like a protective shield. So if a lot of this protein is synthesized, all of DNA is hugged and it's protected. Of course, human wanted to take this technology from tardigrades and to use it for human. So what have been done already? This gene of this protein was inserted in the DNA of human cell culture, and it showed that this protein reduced the 40% the radiation-induced DNA damage. Also, this gene was inserted in the DNA of fruit fly, like a model of multicellular organism, more complex than just cell culture. So what we have seen, the radiation resistance really increased, but there were also some problems. These problems were reduction of locomotor activity and downregulation of RNA expression. RNA expression is responsible for all protein synthesis in our body, so this thing is severe enough, and if we insert this gene in human genome, all human genome, and it will be synthesized for all time, it will be very bad for us. So now we are looking for the middle way to use this protein as a radiation protector, but not to have these severe side effects. So, for example, we cannot protect people just they do not have any mutations. So if we imagine that some mutation will occur on board of long missions, maybe there is another way to solve this problem. Maybe we can just read genome, find this mutation, and repair them by ourselves. So let's look what is done already for this direction. There is a technology of DNA sequencing. It's called Oxford Nanopore technology. You see the picture of the special device. It is like this one size. It's very small. It's the smallest sequencer ever. And it was successfully tested on the board of the International Space Station. Already genomes of Escherichia coli and mouse was sequenced. But how it works? There are two cameras with liquid, and there is special membrane between this camera, and into this membrane are embedded special protein pores. And in this pore, molecule of DNA can go through. And there are also special monitors of electric current. And when DNA goes through the pore, different nucleotides gives different electric current change, as you can see on this picture. So as raw data, we get the different changes in current, and then by artificial intelligence, we get the sequence of nucleotides. So if this technology will progress a little bit, we can detect in real time the mutations on astronauts' bodies. So what we will do further? Further, there is opportunity to correct these mistakes by technology called CRISPR-Cas9. How does it work? It's like scissors. Originally, the system was bacterial immunity. And this bacterial immunity works like that. If bacteria met some virus, it's checked if it knows it or not. If bacteria doesn't met this virus before, it cuts the part of virus DNA and insert it in its own DNA. It's like a special file with different viruses this bacteria met before. And if bacteria met this virus again, it already knows this virus and can very fast fight it. So human took this technology to cut the part of DNA we want and adapted it to give the system the direction. So scientists can just tell the system where to cut, and also they can tell what to insert instead of cut part. So we can just repair mutated part of DNA with the original one. But now this technology has been tested on board of International Space Station, so it works in microgravity conditions, but there are some difficulties still. For example, sometimes this system make mistakes, and also the system have to be given the prepared before special sequences for her to know where is the scissor should work. So if we find the mutation on board of International Space Station, now we have no technology to prepare this special sequence to teach the CRISPR-Cas9 where it should change the broken part of DNA. But this system is already approved to heal people with blood disorders, because blood, it's a really good thing. Let me explain. The problem is this system sometimes make mistakes, but if we take blood outside of the body, then we use this system to repair mistakes. Then we can just check if the system worked well in some cells, take these cells, multiply them, and then put it back to the body. So we are sure that in the body just cells where the system worked well. And people in other countries already get this treatment. What else we have to prepare for our long flights, despite the solving the problems with human body, is the environment. So we have to somehow create the ecosystem which can refresh the water, the air, and food. There are two ways. First, it's full ecosystem approach to create like a little Earth, maybe on the moon or other planets. Now it looks like a really dream. But one interesting project on the Earth was conducted before. I will tell about it a little bit later. And the second approach is simple and compact, when almost all things are solved by machines. And there are just few key plants and bacteria and maybe animals which provide food for crew members. It would be better for spacecraft or maybe for first colonization missions. So the first project which I want to tell you about was started in 1990s in Arizona, United States. They tried to prepare the really full ecosystem inside of a big complex. You can see its real photograph. This complex was consisted from a rainforest, desert, ocean, swamp, and farmland. It was really huge and consisted of more than 3,000 species. And let's look what happened. 8 people lived there for 2 years. They really did it. But it was really a challenge because something happened. They had their oxygen rate fall from 21% as we have here to 14%, like 4 kilometers altitude on the mountains. Why this have happened? Because the ecosystems which were artificially created, they were not stable enough. So the balance of ecosystems shifted just by itself, and scientists could do nothing for changing it back. So people had suffered from the fatigue and oxygen deficit. Also, they lost part of their farming plants, so they suffered from hunger. They used the emergency supplies. Of course, they're not happy for that. So this experiment showed that creating of difficult, complex ecosystems, it's still a challenge. But what happens with this complex now? Now it's owned by University of Arizona, and it can be visited for everyone, for tourists. So if you go to Arizona, you can look at this huge human project. More working idea was the small closed ecosystems. The first one was in Soviet Union in Krasnoyarsk. It was like in just a little bit more than 100 of square meters, where crew members lived for 6 months. They planted wheat and some vegetables, and this system was closed for oxygen and water more than 90%. It's a good result. Can you imagine? Even in 1970s. But they can grow just 20% of food they needed. Other food was dried meals they got from outside. Also, they were second try on the same institute. Then 3 people spent 4 months, and they improved a little bit their system. They built a special machine for burning the plant waste, and also a filter for cleaning the air for better way. So, but this time they could grow more than 50% for their food, but it's still not enough for cosmic mission long duration. After that, Soviet Union broke up, and this experiment stopped. But these results were used during building the next really great project, Chinese one. It's called Lunar Palace, It was performed in 1918. It was really great closed ecosystem. It's how it looks like. The first experiment used one planting camera, and the second experiment used both planting cameras. They really, and here is an image of a plan to use this system on the moon in 1930s, inside of under the moon surface. On this picture, you can see really the vegetables that they could grow in this special place. The vegetables was grown by red spectrum LED lighting. Also, they could prepare the system, which was almost completely closed by water and air. And also, they grew the first try. They grew the 55% of their food, and on the second try, they grew the 80% of their food. So it's the best result ever. The simulation was made, and it showed that this complex could work for 52 years. So it's a really good chance that it will be on the moon just in a couple of years. But why don't we plan to use it on the board of International Space Station or other spaceships? This complex is good on everything, but it needs very much amount of energy, and there is no such amount of energy still on the board of International Space Station. So now this complex for microgravity conditions is a kind of dream. But what have already planted and grown on the board of International Space Station, it's wheat, rice, and chili peppers. Here you can see Soviet experiments. The wheat was grown, and then the harvest was sent back to Earth and planted, and everything was okay. So we know that if we grow something on board of the International Space Station, then we can use this grain to grow on the Earth. Here you can see chili peppers that was grown on board, and the crew members tried them. They were perfect. So it's finish of my talk. I'm happy to answer your questions if you have them. And as you see, future is coming. So thank you.
Okay, please, questions? Thank you for your interesting future presentation. I wanted to ask, well, you've talked about these bears, sea bears.
Water bears. Tardigrades, yes?
Yeah.
Mm-hmm.
And you said that they lived in outer space.
Yes.
More than half the population of it, but then primary population.
They were sent outside of International Space Station, and in 10 days they were sent back, and then they were rehydrated and survived. And astronauts saw that they are survived.
So how they get them back if they were in outer space?
They were just in special, I think, kind of bag or something like that.
And is it outer space?
I think it's...
if they are really small, less than millimeter?
It's a good question. Actually, I didn't check what was it, but for sure it was conditions of vacuum and low temperature, and they were open for cosmic radiation. Maybe they were glued somehow on a surface or something like that.
But so the main purpose was to check the resistance for cosmic radiation.
Cosmic radiation, vacuum, and low temperatures, all of them together.
Okay, thank you.
Thanks for the lecture. And you say that immunity weakening in the cosmos is similar to the immune weakening due to the old age, to the age.
Osteoporosis.
Just a specific issue.
Yes, osteoporosis, it's a losing of bone density. It's like old people have on Earth, but faster.
So, okay, thanks.
So this really looks like that gravity changed time, right?
Thank you for your presentation. I have just about tardigrades. How can it be used in scientific tasks?
Tardigrades?
Tardigrades, yes.
Yes, they are really used. I've read about supercomputer which were built using tardigrades too. I'm not sure what for, but I read about that.
Tardigrades?
Yes.
Supercomputer?
Yes, I think we can find this information after lecture if you want. But for really helpful usage, tardigrade's genome is we search for special adaptations in tardigrade's genome, which gives them the opportunities to survive such extreme conditions. I think it's the main thing that we can understand, and also the opportunity to go to cryptobiosis. So they change their lifestyle from the actively living to almost dried and come back again, and they can do it several times. So it's a really great superpowers.
Okay, thank you.
Thanks for the lecture. With all these facts in mind, especially about bones, we can say that all the talk about flights to Mars is profanity.
No. All my lecture was about that we are going fast to solve these problems, but it's not fast as we want.
Thank you.
Yes, there are problems and they are looking for the solutions. Anybody else? Okay.
And so, what do you think? Would it require for the international cooperation to go to the Mars, or it can be solved on the United States, Russia, China, etc.?
For example, with Elon Musk.
I'm not the expert of this field. I think the international cooperation is the best way to perform this task.
Okay, you said at the beginning, near the beginning, that we need to monitor solar flares in order to protect ourselves, for example, on the ISS. But, well, suppose I'm on the ISS, right? I'm an astronaut. And I get a message from the Earth that a solar flare is coming. And we usually get these messages like a day or two before the flare. Well, we will know for sure a day or two before. And what can I do to protect myself? Well, I'm on a space station and there is no shuttle back to Earth. What can I do?
It's a perfect question. As I understand, right now you can do nothing. But in future, as I understand, there are two ways. First is creation of a really highly protected small space on the board of spacecraft, where astronauts just can go inside and wait for a space flare to come through. And the second way is just to change the course, if it's a spacecraft going, for example, to Mars or something like that.
Or maybe just switch on magnetic field.
Good idea.
Maybe I missed it, but you mentioned there's some explosive material grown in this US biosphere, and it's causing the problem.
The problem?
Yeah, so this bacteria grows?
Yes, bacterial growth. They
So What's the origin of the bacteria?
The soil bacteria, they were 5 meters of soil there inside of this complex. And this bacteria just grow and multiplied really fast.
So how did this Chinese system avoid such
Chinese?
Bacteria? I mean, is this generic?
They didn't have such amount of soil and such amount of different species. All of their environment was controlled, and they used only species they wanted. But the first project was like, we put everything we see around together and let's look what will happen.
I see, it's too much of the mud.
Yes, too much, and they couldn't control it. But here, they almost is controlled, almost everything.
So just they were molded.
Right.
Thank you very much for the talk. I have actually 3 questions. And the first one, then you were talking about experiments about mutations on the 14 or 12 days in space. What is the normal rate of mutations for the people on Earth? Like
It's a good question. I tried to find the answer yesterday, but I couldn't. I think there were no answer in this research, so they did not provide it. But I think the rate is much more high, much more lower. Yes, because the rate of radiation it's 600 times higher on board of International Space Station. So the mutation rates should be something like that lower.
And the second question is like it's quite general about DNA. I can imagine that if you have a photon, like radiation or whatever, it hits the particular molecule, but not all of them. And if you change DNA in one particular molecule, how does it propagate to other molecules? Like if you have mutation in one particular molecule, it doesn't mean that you have it everywhere, right?
No. If you have one cell with one mutation, and when this cell divides, all its children will have this mutation, but not all around cells.
And you also showed the experiment in the very first slide with the blood test on the in space. So why blood is not flying there?
What?
Why blood is not flying?
Why blood is not flying? There is a special, I will show you, special cartridge, this one. It just prepared the way for blood not to fly. Just for blood to be soaked in this tiny place. I think that, I don't know the word, поверхностное натяжение will help.
Surface tension.
So,
Just 2 plates.
Thank you.
So it seems biology is more interesting to this class.
Thank you for your presentation. My question about the psychological condition of the people who participate in experiments in a closed room.
thank you very much. It's a good idea to ask. So, what about the experiment of Biosphere 2? For 2 years, the psychological conditions was not good enough. They start to have conflicts after some time because of deficit of food and something like that. But what about the Lunar Palace station? The reports showed that the first try, one of 3 people felt some stress and even wanted to leave the experiment, but he finished it. And on the second try, there were no problems. It's of course difficult, but this living space is constructed like every crew member have its own room, so they have a place to be alone, and also they had internet to connect with the outside world outside. That's why they try to reduce the stress amount.
Thank you.
Other questions, please?
I have one short question.
vegetation have been on this presentation?
It's Chinese project, yes.
Okay.
Thanks for your presentation. I have a question about CRISPR-Cas9. That technology can cut certain parts of the DNA.
Yes.
But how does it repair after cutting it?
It have, I will show. In the system, it have pre-designed part which it should put back. So there are enzymes which can bind the parts of DNA together. That's how we do it. There are a system of enzymes which bind DNA, and they have a special, like, I'm not sure how.
Like how these enzymes find the specific point.
Specific point by complementarity. I will show you here. You see remember this rule, rule of complementarity. So if this system have a special sequence, it can just put it to the DNA and compare if it fits it or not. So like that, it can find what they need.
DNA is really like a computer code. So, no, I'm not joking. I mean, they are just writing in a sequence, really a program what to do. Right? So, yes.
It's a naive question about the DNA. Is there an importance on this sequence? Well, that should be guanine, cytosine, thymine, adenine, or they can be in random positions?
No. These positions, it's a genetic code. How does it work? So if we make random positions, so we have random information. The three of them, there are every gene consists of a sequence of nucleotides, and they are we can say that they are grouped in three. Like, for example, guanine, cytosine, thymine. These three can code the one amino acid. So we have a sequence of this grouping by three nucleotides, which tells the cell how to synthesize protein with a special amino acid sequence.
So guanine, cytosine, adenine, thymine you said, and thymine, cytosine, guanine isn't.
It's different amino acid.
So it will provide us different behavior.
Different amino acid, different proteins, so different functioning of the cell itself. Inside of the cell there are thousands of different proteins, so we cannot say what will happen like just looking on the code, but if we checked, we can make a kind of database. And then, for example, when we read human genome, we already know the special positions, and changing in these positions will provide some genetic disease. So we can go to the lab and check if we have some genetic disorders, genetically.
Okay, thinking. Biology is interesting.
Yes.
Yes, this is really like a computer file with instructions. So when, say, in physics language, when some devices come to DNA, it reads a sequence and understands what kind of job should be done. So this is really an instruction what to do. So if you change letters, then that will be a random answer.
Yes.
And what about the problems of cloning using this information? People's or some dolly and so on. Can you say anything?
There are some problems. First, I know that in Japan, I think, there is already an opportunity by some $1,000. You can clone your dog or cat if you really want. But there are some problems because our DNA is packed on chromosomes, like X shape, and on the ends of these chromosomes are special stuff. It's called telomeres, and when our cells divide, these telomeres become shorter. So when we become old, really old, we lose all our telomeres, and if we just clone ourselves from these cells, our clone will not live really long, because when we lose all telomeres, we start losing our genetic information. There are some ways to go through this problem, but it's still on research. And also there are problems with epigenetic information, because the information, it's not only this code, but there are special modification of nucleotides, and it's completely more complicated. And it's very difficult to just make the cell to be modified as needed to form from the embryo to living organisms. So it's a really complicated task. And there is ethic problem with cloning people. I know that we can work with embryos, I think for 9 days old or something like that, but not further, because further they are already like people. By laws, they stated that after that they are already people, so don't research them.
In any case, it is better to clone everybody himself in the young age, but not in the, yes?
Maybe there are some tricks, like to preserve the blood from the, I don't know, pupavina. There are special stem cells which can be transformed to any types of the cell. Maybe it is a way. I think that in 20 years we will see something great for that. But already are some sci-fi movies where cloned people live in closed conditions just for organ transplantation. So I think it's also could be our future, and I hope that it will be not.
All right, so other questions, please? Comment? Okay, if not, then let us thank. This was a great lecture.
Thank you very much.